So we've learned that when we breathe in, some of that air will be in the conduction pathway where there's no exchange of gas, while other parts of the air will actually reach the alveoli, and that's where the exchange of gas happens. So how do we calculate alveolar ventilation, which again is the functional part of what we're breathing? So the alveolar ventilation is, you'd have to subtract tidal wave minus the anatomical dead space and then multiply that by the respiratory rate. So how many breaths per minute? Now here again is a summary of the exchange of gases that happens, and we'll go over this all over again. We'll start at the very beginning. This is the air, that's the atmosphere, right down here is the alveolus. And you can see here that the oxygen is going to be transferred through the systemic circulation to the cells, and then there's an exchange of gas that happens here where oxygen is transferred to the cells. And then cells will undergo cellular respiration, producing carbon dioxide. Carbon dioxide goes through the venous circulation to the right side of the heart, where it reaches the alveoli, and there's another exchange of gas where we exhale that carbon dioxide. You'll see here that we intake about 250 milliliters of oxygen, but we exhale about 200 milliliters of carbon dioxide. Now that ratio of the amount of carbon dioxide produced to the amount of oxygen consumed is known as the respiratory quotient. So again, the respiratory quotient is the ratio of carbon dioxide produced to the amount of oxygen consumed. And it depends on our diet. So for a balanced diet, the respiratory quotient is about 0.8, which means that for every 10 molecules of oxygen that I intake, my cells are producing 8 molecules of carbon dioxide. You can see here the respiratory quotient for carbs is 1, while for protein is 0.8, and for lipids is 0.7. So when we intake a balanced diet, we average that to about 0.8. And here again, you could see this whole process happening all over again, where the exchange of gas or the diffusion of gases keep on happening until the different gases reach a state of equilibrium. Now, what are the effects of changes in alveolar gas pressures versus changes in metabolism? So when you increase alveolar ventilation, that means you are increasing the diffusion of gases. There is more intake of oxygen and removal of a carbon dioxide, which is that waste product. While when we change metabolism, remember that metabolism is the source of carbon dioxide. So if you increase metabolic rates, increase metabolism, that means that the cells are producing more carbon dioxide, which also means that these cells need more oxygen. And the opposite would occur in the case of decrease in metabolism. So let's take a look at the different scenarios. What would happen to the alveolar pressures of oxygen and the alveolar pressures of carbon dioxide under different scenarios? So let's say, for example, one person is at this higher than sea level altitudes. They are breathing air with low pressures of oxygen. So the alveolar pressure of oxygen will decrease, but the pressure of carbon dioxide does not change because the carbon dioxide produced by the cells isn't changing. You really haven't changed anything in the metabolism in this scenario. Another scenario would be increased alveolar ventilation with an unchanged metabolism. So now, although the metabolism hasn't changed, but you have increased alveolar ventilation. There is an increase in the exchange of gas. So the amount of oxygen in the alveoli increases while the amount of carbon dioxide decreases. Now, in the opposite scenario of decreased alveolar ventilation or hypoventilation with, again, unchanged metabolism, you'll have the opposite scenario. So the alveolar pressures of oxygen goes down, it decreases, while there's an accumulation of the carbon dioxide levels in the alveoli. Now, in the next two scenarios, we are not going to change anything about alveolar ventilation, but there is going to be a change in metabolism. So the first one would be an increase in metabolic rates, which means that there's an increase in oxygen demand by the cells and an increased production of carbon dioxide, but there is no change in alveolar ventilation. So the amount of oxygen decreases while there is an accumulation of carbon dioxide. The opposite scenario, where there is low metabolism with an unchanged alveolar ventilation, that means that there is an increase in the oxygen because the oxygen demands are lower than normal, and there's also a decrease in the pressure of carbon dioxide because there's lower metabolic rates, not producing the normal amounts of carbon dioxide. That leads to the decrease in the alveolar PCO2. So in order to have a balance between oxygen and carbon dioxide, you want to change both of those variables. So if there is an increase in metabolic rates, there has to be, on the other hand, there needs to be an increase in alveolar ventilation. So, for example, when we exercise, there is that increase in metabolic demands by the skeletal muscles, and that is compensated for by the increase in alveolar ventilation. Think about having maybe a fever where there is an increase in the metabolic demands by the body. To fight the virus, fevers can lead to hyperventilation, and that again is, you want to have that balance between increase in demands and increase in the breathing rates or ventilation rates. Now, before we talk about these two different scenarios, I want to explain what ventilation perfusion matching means. Ventilation means the amount of air that is going in and out of the lungs, and that's obviously related to the patency of the bronchi and the health of the lungs. Now, perfusion refers to the amount of blood reaching the lungs, and that depends on the health of the blood vessels and the health of the capillaries. Now, that match means that a healthy match would be that an alveolus is well ventilated and the blood vessel supplying it is well perfused. Okay, so there is ventilation and perfusion that are equal. That would be a normal, healthy lung, but there are different scenarios where either the alveolus is damaged and that would affect ventilation, or the blood vessels are damaged and that would affect perfusion, and that would lead to ventilation perfusion mismatching. But the lungs have a way to correct that. So we will take this scenario where there is a decreased airflow to a region of the lung. So let us say that a patient comes in with an obstruction in one of, say, the secondary bronchi. That means that anything distal to that bronchus is not ventilated. There is a decrease in ventilation. It means there's a decrease in oxygen in that area of the lung. Now, the blood going to that area, in the capillaries that should be perfusing that area, is going and leaving, but there is no exchange of gas that is happening in that part of the lung because there's no ventilation there. So these blood vessels are going to vasoconstrict and divert that blood away from the part of the lung that is not properly ventilated to other parts of the lung that are healthy. Okay, so again, decreased airflow to that region of the lung due to, say, that foreign object that is lodged into the secondary bronchus. That leads to the decrease in the oxygen levels that will cause the blood vessels to vasoconstrict and divert the blood away. So there's a decrease of the blood flow to that poorly ventilated lung portion to the normally ventilated lungs. Now, the opposite scenario would be if somebody maybe gets a blood clot into one of the pulmonary vessels. That leads to a decrease in the CO2 levels. Now, the alveoli are getting air, but there's no blood for the exchange of gas to happen. So as a reflex, these bronchi will constrict and divert that air away from this part of the lung that is not being properly perfused over to the healthier parts of the lung that are properly perfused. So again, decreased airflow because of that reflex bronchoconstriction, and again, diverting that air away to the areas that have both healthy perfusion and healthy ventilation. Now remember we said that oxygen has to be carried on hemoglobin. Well, this is just 1 unit of the molecule of hemoglobin. Hemoglobin is made out of 4 of these units. Now each unit has an iron molecule attached to globin, and it's also attached to 1 molecule of oxygen. Remember that this is 1 of 4, so a normal hemoglobin molecule will have 4 of these, which means that each molecule can carry up to 4 molecules of oxygen. That would be a fully oxygenated red blood cell. When an RBC is deoxygenated, it would get rid of just 1 oxygen molecule. So it would still have the remaining 3 oxygen molecules, and that means that it is 75% saturated. That is deoxygenated hemoglobin. An oxygenated hemoglobin is 100% oxygenated. You can see this curve right here where when hemoglobin is at 100%, okay, that means that it has all 4 oxygen molecules attached. In the systemic venous circulation, where it is now deoxygenated hemoglobin, it gets rid of just 1 of the 4 oxygen molecules, and it is about 75% saturated. Now we will use this curve. This curve is known as the oxygen hemoglobin dissociation curve. And we will be talking about this curve where how is it that the hemoglobin molecule even knows that I'm near a tissue that needs oxygen, so I need to dissociate. The iron needs to let go of the oxygen molecules. And when you do so, this curve, under different circumstances, for example, if there is an inflammation or an infection in the area, that would shift this curve where instead of being this normal curve that you see, it'll shift it to a curve underneath it, and that is known as shifting the curve to the right. When you shift the curve to the right, that decreases the affinity of hemoglobin to oxygen, and it's easier for hemoglobin to let go of oxygen to the tissues. While the other scenario, which would be a curve above this normal one, and that is known as shifting the curve to the left, that means that you've increased the affinity of hemoglobin to oxygen, and it's extremely hard for that hemoglobin to now let go of oxygen. So we'll talk about these different scenarios. Some of these are normal, and while some of them are pathological. And to do so, we need to understand that in order for oxygen to diffuse to tissues, it has to be free oxygen. So any oxygen that is attached to a red blood cell, for example, we'll take this fully oxygenated hemoglobin that has 4 oxygen molecules. These are not free oxygen molecules, meaning that they cannot diffuse to cells. The hemoglobin molecule, again, has to let go of the oxygen in order for it to be free, and it's these free oxygen molecules that can diffuse to the cells. Now, this whole, these two or three slides are all kind of an introduction on how does the hemoglobin molecule know that it needs to dissociate and make more free oxygen. And here again kind of shows you that the oxygen that is diffused or that was just inhaled through the alveolus dissolves in plasma. Only 2% of it is dissolved in plasma, while the majority of oxygen is carried on hemoglobin. When it reaches the tissues, it will let go of hemoglobin. Now that free hemoglobin can diffuse to the cells. Again, how does this hemoglobin molecule inside of the red blood cell know that it is in a tissue that needs oxygen? And then it will let go of that oxygen. So there are different signals for the red blood cell to do that. So for example, in areas of tissues that need oxygen, there is going to be increased acidity, for example, due to all the metabolic reactions that are being made. Our metabolic products usually are acidic, so low pH or high acidity. It could be if there's an area of inflammation that also, further decreases the pH. There could be a fever. That means, again, that there's an increased demand of oxygen. So all of these factors are going to shift that dissociation curve to the right, making it easier for hemoglobin to let go of its oxygen. Now here are the effects of different things. Okay, so the RBCs have a product known as 2,3-DPG. That stands for 2,3-diphosphoglycerate. Again, it is a product of glycolysis. Remember, RBCs do not have mitochondria, so the only way they can break down glucose is through glycolysis, and they end up making this product, 2,3-DPG. So RBCs normally have high levels of DPG. Now, these high levels of DPG and high temperature levels are going to shift that association curve to the right, making it easier for the hemoglobin molecule to let go of the oxygen. So you will see here that these are the normal levels of DPG in the red blood cell. Under normal levels, the hemoglobin molecule is able to let go. But if you add more DPG, you see that you've shifted the curve to the right, meaning you've decreased the affinity of hemoglobin to oxygen, which means it's easier for that hemoglobin molecule to let go of oxygen. In the absence of DPG, there is a shift of the curve to the left, which translates or means that there is an increase in the affinity of hemoglobin to oxygen, and it is very hard for the hemoglobin to let go of the oxygen molecules. Now this curve shows you what happens in different temperatures. So this right here, the 37 degrees, that's normal temperature, and that is the normal hemoglobin dissociation curve. But let's say in the case of a fever, there is an increase in the metabolic demands of the cells, which means we need more oxygen. So at higher temperatures, that curve is shifted to the right. In colder temperatures, that curve is shifted to the left, and it's harder for the hemoglobin to let go of oxygen. Now you can see here the effect of acidity on that curve. So this is where that yellow line curve is the normal arterial acidity. Under high acidic levels, for example, again, an area of inflammation or an area of high metabolic demands, all of that is producing acid. So in high acidic areas or low pH, that shifts the curve to the right, again, making it easier for the hemoglobin to let go of oxygen, which is needed by those cells. In areas of low acidity, that shifts the curve to the left. Now, a comparison between the dissociation curves between the adult hemoglobin and the fetal hemoglobin. Fetal hemoglobin is a little bit different in its structure than adult hemoglobin. You'll find here that the fetal hemoglobin has a more or a higher affinity to oxygen than adult hemoglobin does, and that is in order to ensure that the fetus gets the oxygen that it needs. Now again, that yellow curve is the normal curve, and we'll see the effect of anemia and the effect of carbon monoxide on that curve. So in anemic patients, there is less hemoglobin to begin with to carry oxygen, which means that the tissues aren't getting oxygen at all, and they are pretty much starving for oxygen. So you will see that in anemic patients, that curve is dissociated to the right a lot, okay, and that means that there is a very low affinity of hemoglobin to oxygen, and it's extremely easy for hemoglobin molecules to let go of the oxygen that it has to the tissues to meet the tissue demands. Now, in exposure to carbon monoxide, the problem with carbon monoxide is that the affinity of hemoglobin to carbon monoxide is 210 times more than its affinity to oxygen. So when hemoglobin finds carbon monoxide, it totally ignores oxygen, and it will combine to carbon monoxide and does not let go. Okay, and that's where the problem is. And this ends this part, and I will see you in the next.